Everyone, this MMI seminar today. The speaker is Lila Kari from the University of Waterloo, who is going to talk about harnessing machine learning for biodiversity informatics and extremophile genomics. Please, Lila, take it away.
Thank you. First of all, I would like to thank for the invitation and giving a talk in this prestigious lecture series. And today, I wanted to share with you our findings of how mathematical structures in the genome used in conjunction with machine learning can be used to find discoveries of biological significance in biodiversity and in extremophile genomics. So I structure my lecture in terms of question and answer. So there are two questions that we are aiming to answer. Question number one is, does biological kinship between organisms induce a detectable mathematical signature in genomes? And the section question two is, can the environment, or at least extreme environment, induce a detectable kinship-independent mathematical signature in genomes? So I will try to answer these two questions with the following contents of the lecture. So I want to discuss a couple of mathematical representation of DNA sequences. Then I want to discuss how one can use those mathematical representation as input for supervised machine learning for taxonomic classification, how one can use the same mathematical representation in conjunction with unsupervised clustering for identification of new species, and finally, how we can use the same mathematical representations to test the hypothesis of an environmental signal in extremophile genomes. So without further ado, I want to explain some terms. So what do we mean by taxonomical classification? Taxonomy is a branch of science that groups and names living organisms based on relationships that are inferred by their shared characteristic. Aristotle, 2000 years ago, was the first scientist who attempted to classify organisms based on how they looked and where they lived. For example, he subdivided plants into shrubs, herbs, and trees, and animals into air, water, and land. Obviously, there are some limitations to that way of classifying. With the Darwinian evolution theory, the possibility dawned that this relatedness is related to common ancestry. And currently, we have a clear picture of what this looks like. So the goal of this entire taxonomic research is to take all life on the planet and organize it into one huge tree of life that originates in the hypothetical origin of life, Luca, the so-called Luca ancestor, the last universal common ancestor, and that groups from where life evolved, and that groups various organisms based on their relatedness and common ancestry. So since this is going to become important later, I want to discuss the major organization of this tree of life. The tree of life has these three major mega branches called domains: bacteria, archaea, and eukarya. So these bacteria are as different from archaea as a polar bear is from E. coli. So all these are major mega branches. And in addition, the eukaryotes are subdivided in 4 kingdoms: animals, fungi, plants, and protists. So currently, to determine whether an existing organism belongs to one branch or another, we don't use morphology anymore. We use DNA-based techniques such as gene alignments for these taxonomic classifications. What I mean by gene alignment, just briefly, you take the same gene that belongs to a certain number of species, the same gene sequence, and then you compute the distance between these gene sequences by the number of insertion, deletions, and substitutions that are required to transform one into another. And then this becomes like a genetic distance, and based on this genetic distance, you decide how distantly related the organisms are to each other. Of course, in the end, they are all related, but you can have like the human and chimpanzee versus a flamingo and an octopus, right? There's a bigger genetic distance. The methods that I'm going to talk about today are not gene alignment. On the contrary, they're going to be alignment free, and this is going to become important later. So before I dwell into the mathematical representations, I wanted to explain why taxonomic classification has become of great importance lately. So this refers to the fact related to Earth biodiversity. Most people don't know, I certainly didn't know before I started working on this, that actually 95%, so this is not a mistake here, it's 9.5, not 9.5, 95% of multicellular species on Earth do not yet have a scientific name and a classification. And actually, since 1948, every single year, people discover 18,000 new organisms, and they're not really necessarily that small. You know, this is just a small sample of the 18,000 new organisms and species that were discovered in 2024. You see this like funny fish, this blobfish, this cheetah-like cipher cat, and so on. And actually part of another project that I'm working on called BioScan, which is a multinational project, we are working on a goal called the Biodiversity Grand Challenge, which is to map all life on Earth by 2045. And why is this specifically important right now? Is because we live in the age of extinction, and actually it happens quite often that when by the time we discover a species, it's already almost extinct. So basically, you cannot protect what you don't know you have. So it is especially important now to really take an inventory and monitor the species on Earth. Therefore, taxonomic classification and identification are extremely important. Okay, so how do we use mathematical structures of DNA to do that? So I'm going to start my talk by discussing one of the main mathematical representations that we use for this research, which is chaos game representation. This chaos game representation, or CGR, this is the only acronym I'll use, of DNA sequences, is a method that was introduced already in the 90s. Recall, a DNA sequence can be viewed abstractly as a sequence over a 4-letter alphabet, A, C, G, and T. And the representation CGR start like that. Actually, I think I'm going to do this with an example. So let's say that I want to plot, to do the CGR of the sequence A, C, G, C, T, G. The chaos game representation starts with a unit square, with the corners labels A, C, G, and T respectively. The first pixel of the square is the middle of the square, of the CGR is the middle of the square. And we start reading the DNA sequence from left to right. So when we read one letter, the first letter being A, we are going to draw a dot in the middle of the segment that is generated by the current point and the corner representing the letter that is being read. So since the first letter was A, the second pixel of the CGR plot is going to be in the middle of the segment determined by the current point in A. The second letter is C, so the next pixel, the next red pixel is going to be somewhere here. The next one is G, so the next dot is going to be somewhere here. The next one is C, which is going to be somewhere here. The next one is T, so the next point is going to be like somewhere here. The next one is G, and so forth. So if you ignore the dotted lines, this like red pixels are the CGR representation of this very short DNA sequence. So as you can notice, one can use this method to plot any DNA sequence of any length. So one is going to obtain just like a cloud of points of, that's going to have different shapes that are going to be the CGR representation of that sequence. So what did I want to say by that? You know, some mathematical facts. So actually, there's a theorem proved in 2015. It's not difficult to prove, that says that given an exact coordinate of a CGR point in the plane, we can determine its corresponding letter in the DNA sequence and its respective position in the original DNA sequence. So actually, in principle, if you have the very last point of the CGR plot, you can reproduce and reconstruct the entire sequence. However, this is only in theory, because in practice, computers cannot store the exact coordinates of all the points, since we are always going to be limited by some resolution. So this brings us to a frequency CGR. So one makes the following observation. So this makes the connection between the CGR generated as a graphical representation and the frequency of words in the sequence. So if you notice here, by the way we plot this, the CGR of a DNA sequence, for example, all the C's are going to fall into this upper left quadrant. All the G's are going to fall into the upper right quadrant, and so on. So if you increase the resolution, you know, all the CC's are going to be in this like a top left quadrant, and so on. So actually one can prove that you can discretize the CGR by some resolution, as there is always going to be some resolution. And depending on that resolution, For example, if you choose a K, if the resolution is 2 to the K by 2 to the K, every so the image of the CGR can be subdivided into little windows, and these little windows, the number of pixels in a little window corresponds to the number of k-mers that of that specific k-mer that occurs in the sequence. For example, let's say that k equals to 3. If k equals to 3, this little square here is going to correspond to the sequence CCC, 3 C's in a row. And so this CGR, this little sequence, corresponds to a matrix where this A11 component of the matrix is 720 CCCs. So this 720 here means that in the original sequence, there are 720 CCCs, there are 203 GGGs, 503 TTTs, and so on. So you see, there is a one-to-one correspondence between discretizing the CGR, or we can compute this as a frequency CGR matrix directly by counting for a given K all the occurrences of a specific subword of length K in the original sequence. So this is the correspondence. So I'm going to talk about this interchangeably, either of a CGR of certain resolution or of the frequency matrix that corresponds to that CGR. So let me give you some examples of computer-generated DNA sequences to start with. So all the sequences that are plotted here have length 150,000. And this is a CGR of a random DNA sequence of this length. So luckily for a square DNA sequence, the CGR representation of a sequence over a 4-letter alphabet actually also looks random. This is the CGR of a mathematical sequence A to the power n. So there is the same number of points, but they are all bunched on top of each other on this line segment between the center of the square and the corner A. This is the mathematical sequence ACGT to the power n over 4. This is a sequence that completely misses A. So you see there is like no point in this corner, the triangle that has corner A. This one avoids CAs, this one avoids TCs, and so on. So we tried actually also CGRs of mathematical sequences that people work, that work on their entire careers in combinatorics on words. So I'm not going to go through the definitions, but you can define people work on Fibonacci words, M-Bonacci words, 2M-Morse words, Lindon words, De Bruijn words, and these are their CGRs. This is a 4-Bonacci word, 2M-Morse word, Lindon word, De Bruijn word. So from the point of view of, visually, aesthetically, this looks a little bit on the boring side. Why is that? Because, for example, the 4-Bonacci word or any Fibonacci word is very periodic, right? So all the points are going to, fall on top of each other based on this periodicity. And the same, these are very periodic, while on the other extreme, the De Bruijn word is almost random. So not that interesting from this perspective. So let's try instead some other CGRs of naturally occurring genomic DNA sequences. You see that this look much more interesting. They are not that periodic. This is a CGR of Homo sapiens, a portion from the nuclear genome, and this is a CGR from an archaeum, which is from the other, one of the 3 domains of the kingdom of life. And actually, these are examples of CGRs of genomic DNA from organisms from all 6 kingdoms of life. This is kingdom Animalia, fungus, plant, protist, bacteria, and archaea. So I just wanted to make some observations. As you notice, first of all, they are interesting. Secondly, they are different from one another. And actually, if you do CGRs of things that are related, the patterns are going to be similar to one another. So the observations that were made already in the late 1990s was that CGR can act as a genomic signature, in that real-life genomic CGRs are species-specific, and the patterns in CGRs are similar for DNA sequences from the same genome. So if you take a sliding window and you plot like a sequence that glides along the genome, the patterns that you are going to observe are similar, but the patterns are different for CGRs of sequences from different species. Moreover, these patterns, the shapes, are preserved regardless of the length and location of the DNA sequence in the genome. So if you take a longer sequence, you're going to get a CGR that is darker usually, and but the pattern is going to be the same. And moreover, if you take your fragment from the beginning, middle, end, wherever, random sequence, the pattern is going to be the same. So these properties qualify CGR for the role of a genomic signature, in that we can use a random small fragment of a genome to classify organisms and to do taxonomic classifications. So, people, especially biologists, they usually don't believe me, but a picture is worth 1,000 words. So this is a picture that summarizes what I said. So these are CGRs of the entire chromosome sequence for all the chromosomes, like 23 chromosomes of the human genome, 1 through 21, 22, chromosome X and chromosome Y. The ones that are fainter because they have more periodicity in them. But you see, regardless of how faint they are, the patterns are the same for all the chromosomes in the human genome. So how do we use these CGRs for taxonomic classification? Well, if you have an input data consisting of DNA fragments, we compute the CGR images for all of them. Then we can use a distance, whatever it is, image distance, matrix distance, whatever, to compute a pairwise distance between all pairs of CGR. And we can use that with a multi-dimensional scaling to obtain a plot. Okay, this is 2D, this is a 3D of the same plot. So we can use multi-dimensional scaling to produce, for example, in this case, like a figure, a 3D plot, whereby each item is represented by a dot or a point in the plot, and the geometric distance between 2 points is proportional to the distance in the pairwise distance matrix between CGRs. So this is a representation, a visual representation of what we obtain by classifying in this way over 4,000 vertebrate mitochondrial genomes. So blue is fish, green is aqua is reptiles, green is amphibians, red is mammals, yellow are birds. And please note, first of all, how little information we have. Like the human genome is 3 billion base pair. This one, each sequence is only 16,000. It's a minuscule fraction of the information in the genome. And it's not, it's coding, non-coding, gene, non-gene, it's everything bunched. And still we obtain this sort of almost spectacular picture perfect classification based on biological relatedness. So the accuracy, if you compute actually the accuracy by using this method hovers around, 80%, low 80s. So we got ambitious and we say, okay, maybe we can use machine learning, we can get a better accuracy. So actually there are two ways in which you can use machine learning. We can add on top of that method that I just described, we can add a layer of supervised machine learning for species classification, or in the case that we don't have information about our data set, we can use unsupervised learning for species identification. So let's start with the supervised machine learning. So the general idea is that the machine learning algorithm is trained. That's why it's called supervised, because you give it to you. It's like having a teacher and it gives you some examples that you can learn from. So we train the algorithm on a training set. In this picture, you say this picture is an apple, this picture is a pineapple. So this is the training. In our case, a training consists of a CGR of a sequence and the species name. You tell it like, this CGR is a fish, this CGR is a bird, this CGR is an amphibian, and so on. And after this training, we test the power of the algorithm to predict the species of an unknown DNA sequence representation. So, you give it a test data and say, you tell me. I don't have any labels, you tell me. This picture, what is it? And the supervised algorithm gives the answer with a certain classification accuracy. So actually for this particular machine learning method, because this is a little bit like plug and play, we used lots of machine learning methods. For this particular one, actually we used a different mathematical representation. So this supervised machine learning we call it MLDSP, which is an acronym for machine learning with digital signal processing. So in this particular case, we used a different mathematical representation where each DNA sequence was converted into a numerical vector. How? You just attribute to every letter of the DNA alphabet a number. So let's say A is 1, C is 2, G is 3, P is 4, whatever. So from a DNA sequence, you obtain a numerical vector, then you compute the discrete Fourier transform of this discrete digital signal. And then one computes pairwise distances between magnitude spectra of these DFTs. For example, one can use Pearson correlation coefficient to compute the pairwise distance. And once you have a pairwise distance matrix, you can use the distance between any given item and all the items in the training set as a feature vector for that item. And then this is the input to and is used to train a supervised learning classifier. So this time the examples are going to be the feature vector between an item and all the other, the distances between an item and all the other items in the data set. This is the feature vector, and the label is going to be animal, bird, reptile. So we did that just to give you some idea of how this works. So for example, this is a digital signal that uses the encoding where T and C are given value 1 and A and G are given value -1. And in this particular one, the blue signal is the DNA sequence from a Canada goose, and the orange one is the DNA sequence of a European beaver. So, what does this mean? So, this means that the Canada goose sequence starts with a T or a C, then the next letter is a A or a G, then the next one is like a T or a C, and so on. So we obtain digital signals. These are the DFT magnitude spectra, and you can compute distances, the Pearson correlation coefficient between these magnitude spectra. So we did lots of experiments with this. We tested several. We test the classification accuracy for many different data sets: the primates, protists, fungi, plants, amphibians, mammals, insects, vertebrate classes, and so on. We also tried many different classifiers in our supervised classification training, and we tried about 15 different digital representations. So here you'll see some examples. So integer, I think this was 1, 2, 3, 4 for the 4 letters, and this PP was 2 letters get 1 and 2 letters get -1. Just A means that one of them is 1 and all the others are 0, and so on. So as you'll see, the best digital signal representation, the best classification was obtained for this purine pyrimidine. So the classification accuracy over all data sets, all classifiers, all different representation was like for purine pyrimidine was 92%, which is a little bit surprising because you would think that you lose some information if you encode 2 letters with the same value. So it's not a bijective DNA encoding, but this is what we got. So the best classifier for these data sets was quadratic SVM, and the best digital signal representation was this purine pyrimidine with the accuracy of over 90%. So notice how the accuracy increased considerably. And actually, in all the experiments that we did after that with many different data sets, the accuracies, if you put supervised machine learning on top of the mathematical representation, the accuracy is from mid to high 90s. So overall, this method results in very high classification based on taxonomy, based on evolutionary relatedness. So this is just some examples of some accuracies. So just look here at the last one. So we compared with other alignment-free sequences. So you see all accuracies are. This is for viruses, 100%, for mammals, 92.7%, for vertebrates, 92.7%. And it's also very fast. Like in the few cases where other methods were a little bit better in accuracy, our method was 0.3 seconds, sorry. And other methods were, couldn't run at all or were over 5 hours. So this proved to be like a very accurate method of classification. I just want to point out again that this method has nothing to do with genes whatsoever. This is alignment-free. So methods based on gene alignment are time-consuming and also require all kind of additional information. So you have to pick a specific gene that starts in a specific location from all the organisms that have that gene. If the organisms don't have that gene, good luck with that. For example, not all organisms have the gene that digest milk, right? The mammals have it, but bacteria don't. So this is like one problem. And secondly, the algorithm is quadratic. So in contrast, this alignment-free methods uses a random small fragment taken from anywhere in the genome, and then it produces this classification of very high accuracy. So this is one of the features of this method, that it's much more universal or generalizable at least. So, okay, so this is so far so good. But I want you to know that supervised machine learning has some inherent limitations, in the sense that you can only predict something the kind of which you have seen before. Like in other words, if you have been trained, if your training examples are all red triangles, and if you are presented with a blue square, the answer is going to be erroneous, predictably, because there is no such class in the training that is a blue square. You see what I mean? Like, for example, if your data set has only animals and birds, and then you give it a reptile, now the algorithm is going to assign, it's going to have to assign a label to this reptile sequence, and it's going to give it by necessity either an animal or a bird. So it's like a predictably wrong answer because it has never seen in training anything remotely similar with the reptile. So these are the inherent limitations of supervised learning, which are even more serious for biodiversity because, remember, we only know 5% of the entire life on Earth. So this means that by definition, our data set is full of gigantic holes. And there is a very high probability that whatever you found is not related to anything that you have in your data set. So supervised machine learning has some limitations. Hence, we also tried unsupervised clustering for identification of new species, specifically to deal with these limitations of supervised machine learning. So, in the big scheme of machine learning for alignment-free genomic classification, you see the one that I talked about before, MLDSPs, belongs to supervised learning, while the one that I'm going to talk about now, which we call Deluxe, is part of deep learning and unsupervised clustering. So methodology-wise, it's a different methodology. Okay, so how does this work? So we call that deep learning for unsupervised clustering of DNA sequences, or IDeluxe. So the mathematical representations that we used for this one is FCGR, the frequency CGR, or that matrix of sequences, or similarly, we can think of it as a discretization of the CGR images. And so the question is, how do you learn? How do you cluster some DNA sequences if you have not been given anything whatsoever but a data set of sequences with no labels? You know nothing, it's like a blind clustering. So how do you find similarities? So the idea here is to seed the clusters artificially. So in this particular case, for each sequence in the data set, we generated what we call mimic sequences. So you have a sequence and you generate a mimic sequence by introducing a couple of mutations in it. So this is an artificial sequence, but it is very closely related to the original sequence. So you generate, for each original sequence, you generate a couple of these mimic sequences and you tell the algorithm, I know you don't know anything about anything, but these three, whatever happens, these three should always be grouped together because they belong to the same algorithm. Okay, so this seeds the iterative process for constructing clusters. So we can use these seeds and the mimic sequences for the algorithm to self-learn patterns in the data. So this is unsupervised clustering. And in the end, you do this multiple times. Of course, there is some variability. In the end, we use a majority voting scheme to determine the final cluster assignment for each sequence. So schematically, the pipeline for Deluxe works like that. You start from some DNA sequences. For each of them, you generate mimic sequences by introducing some mutations. Then you do compute CGRs of both originals and mimic sequences. And you seed the algorithm by feeding it pairs of original mimic sequence and saying that, these two at least that much is true, that these two should belong to the same cluster. So you do this independent training of several copies of the artificial neural network, and then one takes majority voting for a cluster assignment. So this is how this works. And this is like an illustration of how the learning process proceeds. So this is, we try to cluster 2,500 vertebrate mitochondrial genome into 5 clusters, birds, reptiles, fish, those classes. So in the beginning, this is a pentagon, and each corner represents one of the clusters. So this is animals, this is birds, this is reptiles, this is fish, this is amphibian. In the very beginning, each point is assigned to all of the 5 corners with equal probability. But then, and the position of a point indicates the probability that it is assigned to a certain cluster. But then you see by slowly, they start to move probability in this process. And at 150th epoch, everything is perfectly clustered. This contains all the animals all bunched to one another. They belong to the corner Animalia and so on. And as you see here, down here, this is also with a quite high probability. The classification, the clustering from phylum to subphylum, 93%, class to subclass, 85%, 100%, and so on. So this method has also been effective. Okay, so then to wrap up this part of this, the answer to the question, does biological relatedness or evolutionary relatedness induce a detectable mathematical signature in genomes? So the answer is yes. Okay, always is always a difficult word in biology, but I would say that this is, in all the data sets that we've ever seen, we detected this signal. So let me explain here, how do I understand this to myself when I think of this to myself? So what does this mean? Because remember, this mathematical signature is not a certain gene or a certain portion somewhere. It's pervasive across the genome. So the way I think about it is the following. So think of DNA as a language. And imagine that you have two books, one written in English and one written in French, two different languages, right? So assume that I don't know either English nor French, so I have no idea what the books say. However, if I tear out a page from the English book and another page from the French book, and I count all the words of length 3, the English book is going to have a very big spike for the word the, and the French page is going to have a big spike at the word le, L-E-S. So you see, I can tell the two languages apart even if I don't know a single word of English and a single word of French, and I have no idea what those books say. In the same way, we can tell apart two genomes, even if we don't know anything about what they say, what the genes do, and so on, by simply taking a fragment or a page and then counting the k-mers. So this is how I understand to myself, why does this method work and what does it mean? It's a syntactical method that just looks at word frequency. So our next question was the following. Well, so it is this discovery is unexpected because this signature is pervasive, but it's not contradictory to the current dogma. The genome should contain taxonomic information, right? The genome should tell us where things about the evolutionary ancestors of that organism. But our question was, is it possible that perchance sometimes the environment can induce a similar signature in the genomes? So we are pretty sure that the general answer to this question is no. But then we thought, perhaps if you look at some extreme cases of organisms that live in extreme environments, perhaps in those cases we can detect some signature. And this is what we did. We used similar methods to test the hypothesis that in extreme cases, there exists also an environmental signal that is detectable. Okay, so what we studied are extremophiles, which are organisms that are able to live or thrive in extreme environments of temperature, radiation, salinity, and so on. So by extreme, I don't mean that, that this beach is hotter than the other, okay? I mean really hot temperatures in like mud volcanoes of over 100 degrees Celsius, or in Arctic sea ice at -12, or in acid lakes, or in very salty lakes, and so on. So I'm really talking extreme environments. So we focused on extremes in temperature and pH. So for example, our temperature data set has psychrophiles where the optimal growth temperature is less than 20, but in practice, some of them live at -12, or hyperthermophiles where the optimal growth temperature is greater than 80 degrees Celsius, and acidophiles with the optimal growth pH less than 5, so the water has a pH of 7, or alkaliphiles where the optimal growth pH is less than 9. So this is like very salty. So let me give you some more details about the data set. So overall, we collected 700 genomes. So these are the two temperature categories, two extreme categories, temperature and pH. And these are chosen from each of the two branches of the tree of life that I mentioned, of the domain archaea and domain bacteria. Please remember that these are maximally distant evolutionarily. It doesn't get further apart than bacteria and archaea or bacteria and human, for example. Okay, these are maximally divergent. So let me give you some examples. This is one of my favorites, Spirococcus furiosus. So this is an archaeon isolated from heated sediments on Volcano Island. You know, the word says it all. This is a hyperthermophile with the optimal growth temperature of 100 degrees Celsius. It's, you know, boiling water. So the name comes from Pyrococcus, which means fireball in Greek, and furiosus, which in Latin refers to its furiously swimming at 100 degrees. At the opposite end, it's Psychromonas ingrami, which is a bacterium isolated from Arctic polar sea ice. This is a psychrophile where the optimal, you know, it's not that it can survive -12 cold temperature. Its optimal growth temperature is -12, which means it survives even at lower temperatures. So psychrophile comes from the Greek word psychros, which means cold or frozen. For pH, this is Picrophilus torridus, which is an archaeon isolated in a hot spring on Hokkaido, Japan. This is an acidophile with pH less than 0.5. Remember, pH of water is 7. So also, this has a very high optimal growth. So actually, this is a polyextremophile, both acidophile and thermophile, because its environment is both acidic and very hot. So actually, several of these are polyextremophiles. And my last example is another archaeon, which is an alkaliphile, Natromonas pharaonis, found in the Salt Lake Zuf in Wadi Natron in Egypt. This thrives in extremely high salt concentration of pH of 11, which is the equivalent of more than a cup of salt in a liter of water, similar to the salt content of the Dead Sea. So to give you an idea, these are the organisms that we're talking about. So how did we choose the fragment based on which to do our analysis? So our method was the following. We wanted to make sure that the representative fragment that we use is really random. So what we did was the following. We concatenated all the contigs from the NCBI or the genome, you know, into one long full-length genome. And then we randomly selected, let's say, 10 fragments, each of them of a length of 10,000. And then we did a stitch together those fragments in a pseudo concatenation, in the sense that we put another letter here so that we don't create spurious gamers. So let me recap. So instead of taking a single fragment, we stitched together 10 different pieces, each of length 10,000, randomly taken from the genome, and that became the representative DNA fragment of the entire genome, which acted as a proxy of that genome for our analysis. Please notice that this is a very small portion of the entire genome. 100,000 is approximately 3% of the genome length. So it's a very, very small portion of the length of the genome. Okay, so to this we applied supervised learning. So first of all, I just wanted to say that we apply this with many different fragments lengths and many different values of K, okay? So we did a thorough analysis, and it ended up being that the optimal fragment length was 100,000. We didn't need actually 500,000 like in our previous studies, and we didn't need a higher K. K equals to 6, and 100,000 was okay. And this, let's focus a little bit on this result here. So what does the first number mean? In the taxonomic classification, we give the algorithm our data set, and the training set was trained with taxonomic labels, in the sense that we gave it the CGR of a DNA representative fragment, and we told it, this is hyperthermophile. The second one is a psychrophile. The third one is a mesophile, and so on. So the training was with for taxonomy. One is bacteria and one is archaea. And the question was also taxonomic in nature. You gave a new sequence and you asked, is this bacteria or archaea? So you train on taxonomic level, and the answer, the question that you ask, is also about taxonomic level. And as you see here, the accuracy is very high. 99.5% is clearly there exists a taxonomic signal there, because the classification was almost perfect if you train with taxonomic level. Now for the second experiment, we took the very same data set, only that this time of CGRs, right? Only that this time, instead of training with the taxonomy, we trained it with the environmental labels, hyperthermophile, psychrophile, mesophile, and so on. And then the question was also environment. We gave a new CGR of a new DNA sequence and asked, is this a hyperthermophile or what is it? So as you noticed here, the accuracy is also quite high, not as high as taxonomy, but significantly high in order to make us believe that the answer also to the second question is yes, that yes, there exists also an environmental signal, at least in these extremophiles. So we wanted to verify that. So, we wanted to see that, is this real or not, or is it just our imagination? So actually, we said that how about, maybe the algorithm is overfitting or something. Maybe instead of giving it labels and training, just throw all the data set and then just cluster it based on nothing. See based on what criteria does the unsupervised clustering group together organisms. So this is the pipeline of our approach. The input is the data set of bacteria and archaea, DNA representative fragments. And then the first layer of our pipeline is to use unsupervised algorithms to cluster this data set. So most clusters are what you expect. So these squares are archaea and these stars are bacteria. So most clusters are bacteria are grouped with bacteria because their signatures are similar. Archaea are grouped with archaea because their signatures are similar. But some of them are not like that. So some of them are peculiar because this shouldn't happen, right? The DNA should contain information about genealogy, about ancestry, about evolution. But we found in some cases that bacterial sequences or group with archaea sequences based on the black box of the machine learning algorithm. So actually, this algorithm produced 78 candidate bacteria archaea pairs with FCGR that were deemed, based on this algorithm, as similar. But of course, machine learning is a black box. You don't know if they're similar or not. Not to mention that there is a error threshold, right? So you don't know if something is a real phenomena or maybe it's part of the error of that algorithm. So we added 2 more layers to that. So one layer was, okay, let's say that the cluster says that these 2 bacteria and this archaeon are similar. Let's look at their FCGRs and let's compute distances, mathematically compute distances between those, and then make sure that we keep only those that are on a certain threshold. So this is what we did. We chose several distance measures, and we only kept out of the 72 pairs that were output by the clustering algorithm, we only kept 40 pairs where the distance was less than the genus level threshold. To give you an idea of what this means, Canis canis and Canis lupus, the dog and the wolf, belong to the same genus. So we only kept FCGR that was as related as at most the dog and the wolf, not more distant relative. So very closely related. In parallel, we did an analysis of the environmental data. Do they really grow? Do they grow in the same place? Do they grow in similar conditions and so on? So this produced 20 pairs. So in the end, we took the intersection of the 2, and our result was 50 confirmed bacteria archaea. I call them peculiar pairs because they shouldn't exist, frankly, in the sense that they are deemed similar, no matter how we tried to show it, are they really similar or not. They passed all the tests. We added on to that. So these are the groups that we obtained. So we got like 4 different groups. Each of them, for example, this one has a bacterium. Bacterium is blue. Archaeum is in orange. That are both acidophiles and thermophiles. These 2 are both harmothermophiles. So this group should never exist, but they are grouped together as similar, and it seems that they are grouped together based on the environmental factor. Of course, we don't know why, right? But we try to see why are they grouped? What do they have in common? These 2, why should they be grouped together? And when we look at the environmental data, it turns that they are both acidophiles and thermophiles. So this is what we discovered. We followed that up with a statistical analysis. We looked at the overrepresentation and underrepresentation of K-mers for this bacterium and this archaeum, for example, and we found that they look the same, they have the same profiles. And then we did the Spearman correlation analysis. So again, the presence of 3 mers in both of them is similar. So they are really correlated no matter what we try. And actually, our biological team did a fantastic work. They scoured the entire biological literature to see if there is anything that points out that some of these 3 mers are overrepresented or underrepresented in some of this, in each of these organism. So each of these, this is what we found. So what we found is that AGG is underrepresented in this bacterium. And there exists a paper in the literature that says that the amino acid corresponding to this trimer is also underrepresented. So we obtained like a high level of biological corroboration of this. So I present you the crown jewels, at least some of them, that I am completely sure at this point that they have similar CGRs. So one of them is a bacterium, Thermoanaerobacterium thermozaccharolyticum. This is another one is an archaean. Remember how different these are. It cannot get more different in this tree of life. And we also looked visually. So this is the CGR of the bacterium, and this is of the archaeum. So please notice that they look similar. And remember that we compute the distance between them. So the distance between these two CGR is less than the distance between the CGR of a genomic sequence of a dog and a wolf. They are that similar. And then we looked at where they live. And even though they are isolated in different geographic loci, actually they were found later on to co-occur in the same place in Washboard Hot Springs in Yellowstone Provincial Park. So they literally live in the same place. And this is another one, another co-occurring bacterium and archaeum pair, Thermotoga petrophila bacterium, Geoglobus acetivorum archaeum. These are the CGR. You notice that they're almost identical. And even though the isolating environments are different, they are both hyperthermophiles, and they are found to indeed exist in the same spot in the Brothers submarine volcano. So they are hyperthermophiles, and then in the Juan de Fuca mid-ocean ridge flank near Vancouver Island, also a hot hydrothermal vent. So this is what we found. This means what is the biological significance of this mathematical structure in the genomes? So first of all, as not surprisingly, but unexpectedly because of the methods that we use, taxonomy significantly impacts genomic signature composition. So it doesn't only influence this and that gene, it's a pervasive signature that is across the genome. But what we also discovered that extreme environments also noticeably impact prokaryotic genomic signature compositions, which means that environmental pressures are sometimes capable of even overriding traditionally recognized taxonomic influences. So I'm going to stop here for a moment to tell you how I understand this. So not only the environment impacts genome composition, sometimes they override taxonomy. And the way I understand it is as follows, and I'm going to end with this. So you remember that I said that I think of myself as a genome as a book, and the DNA as a language, and word frequencies are the signatures. So this discovery is akin to discovering that actually the DNA, the genome, is not only a book, actually it's a sheet music. And the songs have both lyrics and musical notation. So taxonomy is the lyrics, and the musical notation, let's say you can classify by genre. So you can classify both by language of the lyrics. You can tell so instead of saying that we have two books of two novels, one in English and one in French, you have two song books. One of them has songs in English and one in French. So you can classify them by the language of the lyrics. You know, the lyrics, this song is in English, this song is in French. But if you look at the music, you can also classify them by musical genre. This is pop, this is disco, this is heavy metal, and so on. So this is this discovery that taxonomy impacts genome signature, but also extreme environments impact the signature in the sense that you can also tell the music apart, the genre of music apart. So what I'm saying with this third point here, that environmental pressures are sometimes capable of overriding taxonomy, what I mean that, I'm sure you've had this experience, sometimes the music is so loud that you cannot tell the lyrics anymore. You cannot tell them apart. So sometimes two songs in heavy metal, the instruments are so loud, the music is so loud that you cannot tell anymore if one is in English and one is in French. What you can for sure tell is that they are both heavy metal. Okay, so this is how I understand it. Not always, but sometimes the one of the signature, in this case environment, can even override taxonomic influences. Moreover, these shared genomic patterns persist across the genome, reinforcing the idea that sometimes environmental pressures can shape DNA composition beyond genes and protein coding regions. So, in the end, the answer to my second question, can the environment induce a detectable kinship independent, so music is independent of the language of the lyrics, kinship independent mathematical signature in genomes? The answer is yes, sometimes, and sometimes the environment can even override the taxonomy. So this is what I wanted to say, and I wanted to thank my collaborators, my biology collaborators, statistics collaborators, and my students at Waterloo for this research, and then give you a preview of our next research into multicellular organisms. My daughter has always asked me to look into tardigrades, and I think this is next. Thank you very much for your attention.
Thank you so much, Lila, for this great talk. Are there any questions? You can either unmute or type it in the chat.
Yeah, hi.